Hello students, today we will be studying about antigen antibody reactions. We had studied each of these things separately. Antigens, these are the foreign substances that enter into our body, and there are certain properties which an antigen has to fulfill. Then we studied about antibody. What are these antibodies? These are the substances that is produced by our own body, the plasma cells, and they are produced in response to the antigens, and of course they also have certain properties to fulfill. Now today's class we are going to study what happens when an antigen and antibody reacts, or when they form a complex together. So about them, when do they form a complex? About that we will be studying in today's class. Before that, we need to know about certain things here. We need to know about affinity and avidity. Now what is the meaning of affinity? Affinity means the ability. Now this is the antibody and this is the antigen. Okay, now antigen, I told you, it has certain structures on its surface. They are called as antigenic determinants, or they are called as epitopes. This is the place where the antibody comes and binds. Now this is the antibody. The specific antibody with respect to this particular antigen comes and binds to the. Now this is the place where the antibody comes and binds. That's called as the paratope, or the FAB region. Now, what do you mean by affinity? Affinity means with what there is you know that there is a specific kind of an antigen, and every particular antigen has a specific antibody to which it can bind to. Now, to what an extent the antigen has the capacity to attract a particular antibody, the strength to what an extent the antigen has a strength to combine or the to attract a particular antibody towards itself. So that's called as affinity. What do you mean by avidity? Avidity means after the combination is formed, after the antibody has combined to the antigen, how strong this complex is. Can this be how strong the bonds formed are? Can the antigen antibody, with what force that this complex has to be broken? Can it be broken easily? Can it be should we apply more pressure to break it? So that is the meaning of avidity. So with what strength the antibody is bound to the antigen, so that's called as avidity. Avidity depends on the number of antigen binding sites, or um, sorry antigen binding types, or the paratope of the antibody. So this is the affinity and the avidity. Now let us see what are the different features of the antigen antibody reaction. Now it is specific, means what? It's not that any random antigen can bind to any random antibody. Every time if an antigen has to combine with an antibody, they are specific to each other, they are homologous. It can bind, only a specific antigen binds with that particular antibody. If there are any antibodies in our body already, and if some random antigen enters, because there is an antibody, any antibody cannot come and form a complex with the antigen. The antigen is specific, or every antibody is specific to the antibody, so they are homologous to each other. How is the reaction? The entire molecule reacts with the for the reaction, which means though only the surface of the antigen and antibody are combining, but in the reaction the entire molecule participates when the reaction happens. Denaturation. Now it is, we believe, or it is there is a notion that okay, antigens enter, antibodies go and form complex with the antigen, and the antibody kill the antigen. It is not like that. Antigen alone cannot destroy the antibody alone cannot destroy the antigen. Okay, or it cannot denature, or it cannot destroy. Similarly, antigen cannot destroy an antibody, antibody cannot destroy an antigen. So they both can just form a complex. Now this complex that is formed can in turn attract other or activate other immunological reactions or cells, which will in turn destroy this particular complex. So either one of them cannot destroy or denature the other one. Surface binding. Now what is the meaning of this? Which means the antigen and antibody, or the antibody binds to the antigen only on the surface. They do not penetrate inside the antigen. You can see here, this is the antibody. Antibody has bound only onto the surface of the antigen. It has not penetrated inside. Similarly, you can see this is the antigen. Antigen has not gone inside the antibody. So which means they will bind to each other only on the surface. They do not penetrate into each other. And of course they have to be homologous to each other. Reversible. Now what is the meaning of reversible? Now if you see, this is an antigen antibody complex. A complex is formed if the antibody and antigen are homologous to each other, but after forming a complex also I can detach them. I can reverse the reaction, which means I can separate the two complexes. I mean, I can separate the antigen antibody, and I can separate the antigen. So which means it is a reversible reaction. And what happens as a result of antigen antibody reaction? It will result in precipitation or agglutination and many other reactions. So these are the different properties. So what are the properties? Every antigen has a specific antibody. They are homologous. Only then they will react with each other. Entire molecule participates in the reaction. Though they bind only onto the surface of each other, but the entire molecule reacts. Antibody cannot destroy the antigen. Antigen cannot destroy the antibody. They can only form a complex. Next one is, they bind to each other only on the surface. They do not have the property to penetrate into either one of them. They are reversible. That is, even though an antigen antibody complex is formed, they can be detached from each other. And as a result of the complex being formed, we can see as a precipitation or agglutination reaction. What are the different types of antigen antibody reactions? There are different types like precipitation, I told you, agglutination, complement fixation test, ELISA, immunofluorescence. These are the different reactions that is formed when an antigen antibody is combined. Now this antigen antibody reaction can be in vivo or in vitro. In vitro means in the lab. In vivo means when an antigen antibody complex is formed inside your body. What happens? When do they combine? That is in vivo. What and all happens inside your body when they both combine? So that is in vivo. And if these reactions are happening in the laboratory, then it is in vitro. Now in vivo, what are the different types of reactions? Neutralization, immune complex formation, opsonization, complement fixation, and AdCC. Okay, the first one is neutralization. Now this neutralization generally happens if it is associated with a virus or maybe a toxin. Then neutralization happens. Now all these reactions, whatever we are talking is which is happening in our body. When an antigen enters into our body, of course an antibody is produced and some reaction happens. So about that we are going to study. First kind is neutralization. Means it's either when this neutralization gets activated when either a virus is acting as an antigen, or a toxin is acting as an antigen. Now here what happens is, I had told you that antigen has many antigenic determinant sites. Antibody has a minimum of 2 antibody binding sites, antigen binding sites or paratopes, depending on the kind of antibody. So what happens in neutralization is, So the virus is there or the antigen is there. Now, the antibodies come and block the antigen completely. So, when the antibodies completely come and block the antigen, what happens? Antigen has no free space or free receptors for itself to go and bind or attach itself to any part of our body. I told you any antigen can cause a disease. First step is it has to attach. So if the antibodies come and block the antigen completely like this, there is no receptors that are free, and hence those they are completely blocked, a complex is formed, and hence the neutralization happen, and hence the antigen is not able to cause any disease to the host. So that's called as neutralization. Opsonization or opsonin formation means it is something like formation of a bridge. It generally happens during, I'll give you an example of a complement system. A complement system is it is made up of many protein fragments, and this protein fragment goes and attaches to the antigen antibody complex, and ultimately this as a result of it will kill the antigen. In detail probably we'll be doing when we are studying about complement. Now here what happens is there is a bacterial cell, here, and there is an antibody. Now what happens is the antigen is there, antibody is there, a complex is there. Now the antibody has to this part, the different parts of the antibody. This is a region where the antigen it can bind to the antigen. Now this is called as the FC region. This is called as the now this part of the antibody is called as the FC region. FC region means to that part the complement proteins can come and bind. So many proteins will come and bind to the FC region. Now what these complement proteins will do is it will activate the phagocytic cell or the macrophage. Now what it will do is the macrophage cannot directly come and bind to the antibody sometimes. So the phagocytic cell will get attracted by the complement proteins here that is formed. So where and the macrophage comes and binds to the complement protein. So now who is acting like a bridge here? The complement protein. Now here you can see there is a bridge. This is the antigen antibody complex, this is the complement protein, and this is the macrophage. So this macrophage ultimately comes and binds to the complement proteins, and then ultimately it will engulf the entire complex. Now suppose if this complement proteins were not there, this phagocytic cell may not have got attracted to this complex, and definitely will not get engulfed, and this complex would not have got denatured. So this the complement proteins, this is acting like a bridge. Now you can see here this is a complement protein. Now this is having a bridge, this is building a bridge or connecting between this complex and the phagocytic cell. In this picture it may be more clear. Now this is a pathogen, this is the antibody, and to the antibody a bridge is being created, that is the fragment, the complement protein. To the complement protein the macrophage is coming and attaching, and finally the macrophage engulfs this entire complex. So that is about opsonization. The next one is again your complement itself, whatever I was telling you, the complement that is getting activated, either it will stop at one particular point and then attract the phagocytic cell, or the complement, this is the antigen, this is the antibody, this complex in turn brings in the complement proteins together. You can see many complement proteins are forming here like a chain. Ultimately what happens is these complement proteins will make many holes in fact, in the antigen. Now because the holes are made, what happens? The cellular components are leaked out and ultimately the cell is dead. So that is about your complement, how the complement actually kills the uh, antigen. Now how this complement gets activated? Only because this antibody was there. It's not that complement it gets activated only in the presence of antibody. This is one of the method. In the absence of antibody also complement can get activated, but antigen antibody complex also will activate the complement. So this is one of the ways that the antigen can be killed. The next one is AdCC. Now here what happens is, basically this happens during your allergic reactions. When your IgE, generally your IgG and IgM gets activated, or they are produced when there is any reaction or when there is any antigen that is and that's entering into your body. Now due to certain genetic disorders, what happens, instead of IgG and IgM, IgE gets activated. Now this IgE goes and attaches itself to the mast cells. Okay, now when they go and attach themselves to the mast cells, what will happen is, I am telling this in brief. In detail we will be doing this when we are studying about the hypersensitivity reactions. Now what happens here is, or the natural killer cells, anything it could be, the antibody is there. Which antibody? IgE. Antigen is already bound to the to the antibody. So the red one, what you can see, they are antigens. It should have been IgG antibody, but instead of that IgE gets activated, so IgE is there. Now this antigen antibody complex IgE, because it is IgE, it goes and attaches itself to the histamine, natural killer cells, okay, or the mast cells. Now when they go and attach to the mast cells, it will result in dilation of the wall of the mast cells or the natural killer cells. As a result, the cellular components are released out. Now when the cellular components are released out, what comes out? Generally histamine. Okay, histamine is released out, histamine and heparin. Now what is histamine and heparin? They are allergic. Now these histamine and heparin can cause any allergic reactions, like probably they can increase the activity of the goblet cells, they can increase the contraction and relaxation of your muscles, like your lungs. That's your there is a particular level of contraction and relaxation of your muscles at a particular speed, but this speed increases, so that is why the person goes breathless. The 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 level of breathing increases there. There is excess secretion of mucus. There are various other things that happens because of the excess secretion of histamine. Now where the histamine is released out, I had told you. So that is AdCC. So this is one type of reaction that happens. So these are all the reactions that happens inside your body in vivo. In vitro also reactions happen. That is, there are different things that happen. Precipitation happens, flocculation also happens. Now what is this? If there is a soluble antigen and antibody, okay, and this soluble antigen and antibody forms an insoluble complex, okay, and this happens in the presence of electrolytes at a particular temperature and pH. This is called as precipitation. Now this entire thing, whatever I told you, if the complex that precipitate, if it instead of settling, if it remains suspended in the entire solution, it is called as flocculation. Generally when you do your slide test, you get flocculations, and whenever you do your gel diffusion test, that you get precipitation reactions. Now what is a precipitation reaction? Now how can you see a precipitate? When you mix an antigen in the lab, you take some amount of antigen and its homologous corresponding antibody. When you mix together, you can see a complex, you can see some kind of precipitate. Now how can you see that? This particular thing happens because of certain reasons. Now why is that, how this happens? It was actually explained by Marak, and this is called as lattice hypothesis. Now let us see what it is exactly. I had told you that see, when an antigen combines with an antibody, it will form a complex. Now this antigen antibody cannot be present in any amount. It has to be produced in such an amount that it has to, all the antibodies has to be interconnected to all the antigens present. Okay, no antibody or no antigen should be let loose. If either one of those are let loose, then you cannot see a complex. Probably they will form a complex, but you cannot visibly see them. Now suppose for example, you know that antibodies generally they are bivalent, and antigens they are multivalent. They can, many epitopes are there, and paratopes for an antibody are of 2 for a general antibody. Now here what happens is, say you can see this, what I have drawn here, what I have represented here is the antibody, and whatever is shown in dots is antigens. Now suppose if there are more antigens and less of antibodies, you can see that antigen is combining with antibody, but all complex is not formed, they are still left free. So this is zone of where there is more of antigens, you cannot see the precipitate. In this particular scenario, if you see, the antibodies are more, the antigens are very less. So in this particular scenario, what is happening is, again, some of the antibodies are left loose, they are not connected. All the antibodies should be connected to all the antigens and interconnected. Nothing should be left empty there. Okay, now here they are left loose, so again you cannot see a visible precipitation. So this is zone of excess of antibody. Now here you see, I'm not telling that they are antigen is equal to antibody, but it's called a zone of equivalence because all the antigens present and all the antibodies present are interconnected to each other. So that is called as zone of equivalence, and when such complex is formed, you will be able to see the precipitation reaction. So you can see, represent this in the form of a graph. When there is excess of antigens, again you cannot see the it may be happening, the reaction will happen, but you cannot see it. Reaches the peak, the reaction reaches the peak when there is equal amount of antigen, I mean when the all the antigens combine with the antibody, and you can see the precipitation reaction. Later on, the amount of when there is excess of body, again you cannot see, maybe the reaction is happening but you cannot see because one of them is excess there. So this is the lattice hypothesis which was told by Marak. So if they ask you about the mechanism of precipitation, you have to talk about this lattice hypothesis. Now if you take a test tube, in a test tube, suppose there is an antigen here, suppose there is an antibody here. Now here also you cannot see the precipitation, here also you cannot see the precipitation. Slowly, as the antibody tries to move inside, or the antigen goes towards the antibody because of the attraction, you can see a precipitation ring that is formed at the junction of the two, because at that particular place a complex is formed, and that is why a precipitation reaction, a ring can be seen there. So this you can say as a ring test, okay, or a tube test also. Other two places you cannot see the precipitation because here antibody is more, here antigen is more, definitely you cannot see the reaction here. Moving on to the difference between the precipitation and the agglutination, I had already told you, I don't have to go into the details of this again. Here if I have to show you the difference pictorially, precipitation and agglutination. So this is how precipitation and this is how the agglutination actually looks like. Okay, So this is again the antibody, the antigen, and the complex which is formed, the lattice hypothesis, and this is the definition of it. Now, precipitation can happen in liquid or in gel. Now, if it happens in a liquid, this is how the tube test, the tube reaction. You have the antibodies here, you have the antigens here. Both the places you cannot see the reaction. But when they start moving towards each other at one particular junction, you can see a precipitin ring. So that is called as the tube test, where you can see at this particular point, you can see that the antibodies are more, this part wherein the graph is inclined, but here at the peak the complex is formed, and as you go up again, and the other component, the antigens are more, again it reaches to the down and you cannot see the complex. Now, gel diffusion technique, you can divide them as double diffusion and single diffusion, radial immunodiffusion technique, and Octerlony double diffusion. Now, radial diffusion, here it is excuse me, it's a single diffusion. Now, single diffusion means, see, there is an antigen, there is an antibody, one of the component is diffusing here. Okay, so that is what here. Now, when do we do this radial immunodiffusion, or RID? This I know what is an antigen, I know what is an antibody, but I want to know what is the concentration of an unknown antigen that I am using here. So in that case, I will use this particular technique. So what do I do here is, I take a plate, and into a plate I will prepare 1% agarose gel. Now, why do I prepare agarose? Agarose is because it has high penetration power, and into the agarose, when I am preparing the gel, I will add 1%, that is agarose I will prepare, I will add, and into that I will add the antibody. Okay, now the antibody is added into the agarose, still it is in the molten form because it is hot. Then when it is in hot conditions, I will pour the agarose into the Petri plate. Now, after, remember, I will allow it to solidify, means I will cool. Now, after cooling, what will happen is the antibody is what do you say, it's inside the agarose. Okay, now it cannot move, it's inside the agarose. Then what do I do is, I will place I will make holes, I will drill holes, or I will drill wells into the agarose. Now, after making wells into the agarose, around 4, 5 or 6 wells I will make. Then what I will do is, into the first well I will add antigen of known concentration. Okay, and into the second one I will add again another antigen of another known concentration. The third one I will add antigen of another known concentration, and this I will add antigen of another known concentration. So same antigen is added, and I know all these antigens, which antigen it is, and I know these antigens are specific to the antibody that I have added into the gel. And I will add the fifth antigen into the another well. I don't know what is the concentration. Then I will keep it inside, I will place a cotton which is moist, and I will keep it for incubation. Next day I will come and see the result. Now what will happen is, see, the antibody is inside the gel. Okay, it will not move, it will not diffuse. The antigen is there inside these wells. What will happen is, the antigen will start diffusing into the gel. I know the antigen is specific for that particular antibody. So according to the concentration, the diffusion level, if the concentration of the antigen that I have added here is more amount of diffusion I can see. Now how do I know it has diffused more? The precipitin ring that is formed will be bigger. Okay, and if the precipitin ring is smaller, which means the antigen concentration is less. So smaller ring that is formed around is because lesser concentration, bigger ring means more concentration. Then I will calculate, I will take the diameter of all the precipitin rings that is formed, antigen 1, antigen 2, antigen 3, antigen 4, and then I will correspond, I will take the measurement of the ring of the unknown antigen. and I will compare this unknown antigen diameter with the other diameters. To whichever it is corresponding, I will say that this particular antigen has the same concentration of that corresponding ring antigen that was added. Suppose if this unknown antigen ring is say 0.3 mm, and the antigen number 2 that I've added is also showing 0.3 mm, then I will say the concentration of these two are the same. So that's how I will find out the concentration of the unknown antigen. So this is about radial immunodiffusion. So till here we will study. The other techniques we will study in the next class. Thank you, students.